Okay.
So we can start now.
Okay. Thanks, Colleen. So we have a great turnout, and I'm sure people are going to straggle in the next couple of minutes. It is really my pleasure to introduce Dr. Soumya Raychaudhuri as our speaker today. Soumya did his MD PhD at Stanford and then moved to Harvard and has been there ever since. At Harvard, he did at Brigham and Women's an internal medicine residency, followed by a fellowship in rheumatology, postdoc with Mark Daly, and then moved quickly through the ranks and is now a professor at Harvard, a medical school director of the Center for Data Sciences at Brigham and Women's and Harvard Medical School, an associate member of the Broad Institute. And the reason why I invited Soumya is really, he's really leading the field in my estimation of folks who are integrating genetics, genomics at the single cell level and with molecular biology. So we're very pleased to invite Soumya to speak on covariant network analyses in single cell, and he's encouraged folks to raise their hand or type questions in the chat, and Colleen and I will interrupt and tell them if we see a question. So Soumya, all yours.
Okay, great. Let me go ahead and try and share my screen here. So can everyone see everything?
Yes, perfect.
Yeah, great. Okay. So today I'm going to talk and thank you, Judy, for not just for having me here, but also for that really nice introduction. Today I'm going to talk about strategies that we've been taking to define disease-associated single cell states. And I'm going to talk specifically about covariant neighborhood analysis. The co got cut off there. And just to give kind of a little bit of background about myself and about how I got into single cell genomics. You know, my lab for a long time has thought a lot about autoimmune diseases, particularly the genetics of autoimmune diseases. And we focused a lot on rheumatoid arthritis. I'm a rheumatologist, and rheumatoid arthritis, as many of you know, is an inflammatory disease where you have inflammation of the joint tissues, in particular the synovium. And Pierre-Auguste Renoir had that disease, and you can see his hands were really severely damaged by this condition. Despite that, his self-portrait still looked like the one here, even when his wrists were too damaged to hold a brush in his hand. Yeah. Sorry, this is really weird. This is not this talk I thought I was giving. Okay, I see there's some, sorry, apologies, some hidden slides. So, one of the ways in which we've been thinking a lot about RA and other autoimmune diseases is trying to use genetics to find all the different causal alleles that drive RA susceptibility. This is our Nature paper from 2014 showing more than 100 risk alleles associated with RA. And the more and more of these alleles we start to find, the more we realized it was really important for us to understand what those alleles were doing and what cell types they were acting in. So once you identify a disease SNP, you know, we, like many in this field, including the inflammatory bowel disease group, has been very interested in thinking about how these alleles alter gene regulation. And of course, many of us think that a lot of these SNPs, the causal ones at least, live in regulatory regions that are driving changes in gene regulation. And they're probably acting in specific cell types of interest. And so, you know, we, like many people, started thinking about expression, quantitative trait loci. We started thinking about editing SNPs to try to identify whether they have specific effects on gene regulation. But of course, all these things have to be done in cells and have to be done in the right cell types with the right cell states. So we recognize that RA risk alleles drove T cell gene regulation in general. And so hence it became really important for us to understand what those key T cell states in RA were. And the strategy that we took, like many in the field, is to start thinking about human blood and tissue samples of individuals with RA and controls, and then started to ask, what are the cell states that are most associated with disease, the T cell states? And if we could find those, maybe those would be the cells that we'd be most interested in making sense of. And we were, of course, abetted, as I mentioned, that last genetic study came out around 2014, and that was right at the beginning of the single cell revolution. This is a kind of a common slide that I'm sure many of you seen, which is the smoothie slide that, of course, only a few short years ago, we were all looking at bulk RNA-seq profiles or bulk microarray profiles, which were averaging many cells together. And really what we were looking at was a smoothie. And actually what we want to do is we want to look at those individual cells. We want to look at the individual grapes, the individual bananas, to understand what those alleles are doing in those individual cells. And there were multiple technologies that I'm going to make allusion to. Some of our early work thinking about single cell association studies were focused on using CyTOF data, which of course is a heavy metal labeled antibodies based strategy. And the cells are, instead of flow cytometry, we're staining cells with antibodies with heavy metals, and we get data on as many as 50 or more channels for every individual cell. And then, of course, more recently, the single cell RNA-seq profiling has really taken off, has become, affordable is probably too strong of a word, but has become really possible to be applied widely with the advent of 10x genomics and other related technologies. So when we think about cell type, cell state heterogeneity, and single cell technologies, there are, of course, many challenges we think about too. And in particular, one of the things I think we all know is that sample quality can vary dramatically from batch to batch. Signal quality can vary from cell to cell, even within the same batch. And that makes it hard to define individual cell populations. And that makes it really hard sometimes to associate populations to disease state, because all of these challenges are compounded. And so the simple question of what are my cells, what cell states do I have, and what are the cell states that are most relevant to disease is confounded by each of these different variables. So I'm going to tell you about our journey along this way and some of the ideas that we've come up with to address this statistical and technical issue, and try to show you some examples of successes along the way, and hope that this will engage all of us in a discussion, because I'm sure your group is thinking about some of the same things. So first, I'm going to talk about what I call cluster abundance methods. I'll talk about our own cluster abundance method, but there are others, and I think they have similar merits and demerits. And then I'm going to move on to talk about covariant neighborhood analysis. In the first part, I'm going to talk about some applications to rheumatoid arthritis, blood and tissue. In the second part, I'm going to talk about some applications to blood, looking at T cells from tuberculosis patients. So what do I mean by cluster abundance methods? Well, so the strategy here is pretty straightforward. The idea is maybe you have all your single cell data, whether it's from CyTOF or single cell RNA-seq, and you can cluster your cells. And in this case, your cell state definition is basically the clustering that you get out of your clustering algorithm. And then you can simply ask, well, are my cluster frequencies different between cases and controls? And then you can assign a statistic. In the most simple case, maybe something like a t-test. But that may or may not be the best strategy, because you might need to account for covariates, because you can imagine tissue quality, sample quality, other clinical factors such as age, sex, et cetera, might influence cluster abundance as well. So we started thinking about this topic around 2014 or so. And we started thinking about it for the first time with CyTOF. And at that time, there was tons and tons of methods to cluster CyTOF data, just as there's now tons and tons of methods to cluster single cell RNA-seq data. But so we recognized the real challenge was to think about how do we associate clusters to individual disease states. So it's critical to not only be able to identify and define these key populations, but also to identify phenotypes that are enriched for them. And the challenge, of course, was, as I mentioned, batch effects, intra-individual effects, technical effects that might actually affect the propensity for a cell to end up in a batch or for some or in a cluster. So some clusters may be bigger or smaller, but not for the reasons that we care about. So when we first started thinking about this, we thought, okay, can we approach this in a very simple way, a naive way? And the most simple thing you could imagine doing is that you take all your cells, put them together. You can define a TSNE plot or a UMAP or something like that. And then you can ask what % of my cells are in a certain cluster versus not. You might imagine here we're looking at, say, T cells and the landscape of different T cells. And you can see in this toy example we have that you have a difference of 9 versus 3%. That's a threefold change. You can do a t-test. Now, the problem, of course, is that these clusters aren't super reliable. And then the second problem is estimating cluster frequency can often be pretty inaccurate, right? Especially if your clusters are very small or if you have a small number of cells for a cluster in a given sample. That makes these frequency estimates unstable. Of course, when we do flow cytometry or something, this is a very standard approach. The other thing that I continue to see in the literature that I'd encourage all of you to avoid, but is a strategy people often use, is that you treat every single cell like it's its own event. So you might say, okay, I've gotten a data set of 20,000 cells or more. How many are in the cluster that are in case samples? How many are in the cluster that are control samples? This approach basically assumes that every cell is an independent observation. It ignores the fact that the cells from the same person are more likely to be similar. So the beauty of this, if you're writing a grant or something, is you can get an astronomical, very convincing p-value. The downside is your data is likely not to be reproducible. So as we started thinking about these different challenges, we came up with a strategy called MASK, or mixed effect analysis of single cells. And the idea here is that you have a cell and you're trying to predict how likely is the cell to end up, in a particular cluster. And you have a linear model where you have a fixed effect, which might be clinical variables such as sex or age. They might include technical variables such as read depth, but they also include random effects such as what's the donor, right? So if you have multiple cells from the same individual, you want to take that into account with the random effect. That's your baseline model. And then you ask, okay, if I understood what the case control status of an individual is, can I improve that prediction? And that was the approach that we went with. So, and this work was done by Chamit Fonseca, formerly a grad student in my lab. And as you can see here, we were thinking again of looking at blood samples from case and control individuals. And this is a little bit of the math. So what we're doing is predicting how likely a cell J is to end up in a cluster S. And then we have an intercept term, which kind of gives you a sense of how big the cluster is. Then we have clinical and technical variables, which are variables which result in a cluster being bigger or smaller. So for example, if aging has an effect on T cell frequency, this is where that would get taken into account. And then we've got our random effects, both technical effects for batch as well as inter-individual effects for each person. And then finally, we compare that baseline model to a model that includes a case control term. So we ask how much do our predictions of predicting whether this cell ends up in this cluster, how much is that improved if we add this term? So, all right, so we want to make sure this worked well. So we had some side-top data. We clustered the data. We defined 26 cases and 26 controls. And then what we did was we randomized case control status. And then we applied this model. And the good news was that about half the time we saw p-values of 0.5 or less, 10% of the time we saw p-values of 0.1 or less, 5% of the time we saw p-values of 0.05 or less. So that told us that this model is pretty reliable. That binomial test that I told you, where you treat every cell as its own event, or a chi-square test, as it's commonly done, you can see the problem with that approach. If you do that even in random data, you see that half the time, you can see that almost in 50% or more occasions, you get a p-value of 0.01 or less. So it's a hugely problematic way to do things. So we put this model to the test, and we did a study where we obtained blood from RA cases and RA controls. We teamed up with our colleague Michael Brenner and Deepak Rao, who's now an assistant professor, but then was a postdoc in Michael's lab, and Chamath and Nick Teslovich, who worked out all the CyTOF in my lab. We wanted to say, can we define what the CD4 T cells are in RA patients? What are the T cell populations that are associated with blood? Which populations and states are actually expanded in RA? Again, with the idea that if we could understand what these T cell states are, maybe they would help us understand something about disease. So for this study, we use mass cytometry, and a little bit later on, I'll be moving to single cell RNA-seq. We designed a panel of antibodies to query CD4 cell lineage, activation, and exhaustion. We queried, in this case, we looked at T cells both before and after stimulation. And so we obtained our data, and as you can see right away, we saw a beautiful spectrum of T cell states. So looking up here, you see the effector versus central memory T cell divide with CD62L. You see the Treg population, that's CD225 positive, FOXP3 positive. We stimulate the T cells, and we largely saw a lot of the same clusters, though obviously some of the staining had changed. In each case, we defined about 20 clusters. So now we could go through, and for each of these populations, we could say which ones are increased in the disease state using MASK. And we found this one cluster, cluster 18, both in resting and stimulated T cells, which are these DR positive CD27 negative T cells. I'm not going to dwell too much on their function, but they came out of this analysis. And you can see the kind of statistics that you get from an approach like MASK. You see an odds ratio, so it suggests about a twofold expansion. It's statistically significant. And again, we want to be sure that whatever we're doing was sensible. So what's more sensible than, because we knew this cluster was defined by two markers, we could go back, get independent samples, sort out T cells, stain for CD27 and HLA-DR, identify the T cells that are positive. And indeed, we saw that this population was about twofold expanded in RA compared to controls in these 66 independent samples, exactly consistent with our expectation. So CD4 T cell populations, this particular population was expanded. This DR positive CD27 negative T cell population was seen in the single cell CyTOF data. We had to use this mixed effects model to find this expansion. I told you that it was about twofold expanded in blood. We also observed that this population was 5 to 10-fold expanded in synovial tissue. And this population had features of chronic stimulation. They had Th1 skewing. They secreted a lot of interferon gamma. Again, I won't dwell on those results because I want to get to the other strategies we use to define associated populations of cells. These approaches,
Shamo, before we go on to the tissue, you want to read the two questions in the chat. The first one is from Shifa. Do you have any reference sample for this model?
So for this model, no. So this is sort of designed to simply be able to take cases and controls and simply do the testing. The clustering is done on the samples themselves. We have another algorithm, for a lot of the single cell RNA-seq work that we're doing now, we use an algorithm called Symphony, which allows us to map cells to our reference. So sometimes we cluster things de novo. Sometimes we're interested in assigning cluster identities based on a reference data set, and that's the way we do it.
Okay, and the question from Lucia, what are the advantages of your method compared to comparing cell frequencies in a given cluster for each donor using a linear model, considering that mixed model may be heavy in terms of computational resources?
Yeah. The big advantages are that you actually have more power. And again, I didn't show these results, but you actually have more power than doing a simple direct comparison. Part of that is because you're able, so for example, if you had a very small number of cells from a sample or a smaller number of cells from some samples and a bigger number of cells from other samples, your ability to estimate those proportions is much more accurate in the bigger data sets. In addition, this model allows you to correct away a lot of technical factors, some of which are single cell technical factors. So for example, when we're doing an association using single cell RNA-seq data or CyTOF data, we can use some kind of a parameter for data quality. So for single cell RNA-seq data, that might be, for example, read depth, right? So you're more confident about a cell where you have greater read depth in terms of where it ends up in the cluster. And this model is able to kind of account for that stuff.
Great.
Okay, so can this model, we talked a little bit about it being used in blood. I'm going to just touch a little bit on its use in tissue. So this is a large consortium effort called the AMP Phase 1 project, or the AMP project, Accelerating Medicine Partnerships Consortium for Rheumatoid Arthritis, joint consortium involving a lot of different sites. And this was the Phase 1 paper that we did. In this paper, we were very interested in understanding what those driving populations in tissue were. And here we obtained synovial biopsies from about 10 different clinical sites. The tissues were disaggregated, analyzed with mass cytometry, analyzed with bulk RNA-seq, and also single cell RNA-seq. Now, in this particular study, it was in the early days of single cell RNA-seq. So we didn't have enough single cell RNA-seq to do any kind of association testing, but we did have a lot of tissue mass cytometry data. And again, our mask strategy was used here to find populations that were case control associated in a way that was much more accurate and better powered than doing sort of population frequency testing. I'll just show one example. So this is a monocyte population. So when we looked at our inflamed RA samples on the far right versus OA and non-inflamed RA samples, we saw this like massive orange population. Here are all the different populations seen in mass cytometry. And you see that bottom population is indeed about 8-fold expanded. With the single cell RNA-seq data, we were able to see that this was an IL-1B positive population that seems to be expanded in synovial tissue inflammation. We went back and looked at the tissue. And indeed, if you see this staining for this population, and it's the green that you should be looking for here. When you compare RA to OA, you see a huge difference. And again, you can blow it up and you can see that green IL-1 is coming from exactly those culprit cells. So let me talk a little bit about cluster abundance methods before I switch gears and start talking about covariant neighborhood analysis. So cluster abundance can be a really effective way to identify cell states that are disease associated. It's important to consider donor effects as we do with these mixed models. It's a linear model that, again, allows us to also account for these technical effects like read depth or data quality. And I mostly so far have been talking about application to CyTOF. And we were able to identify a bunch of RA-associated populations that were functionally validated, but there's some possible limitations of these cluster abundance methods, which will bring me to the next part of the talk. And these are probably problems that are pretty obvious to anybody who's worked with any kind of clustering algorithm. The first is that cell states aren't clusters, right? So when we define our clusters, we're often taking a continuous space of cells, right? And we're partitioning them into clusters. And some of those boundaries can be very arbitrary. And some of those boundaries can be very sensitive to parameter choices. So for example, choosing different resolution parameters result in more or fewer clusters. The other thing which is complicated is when we test for association, you're testing the clusters individually, but you could imagine that that the key driving population is smaller than the clusters or is actually covering multiple clusters in aggregate. So that might be reducing our ability to find the populations that we kind of care the most about. So that brings me to
Shomo, before you hit neighborhood analysis,
yeah, please.
2 additional questions. Shifa, do you apply downsampling with mask?
So the answer is that the way we do it is to define the clusters. Sometimes we do downsampling because we worry, as with any clustering algorithm, that if you have too many cells from certain individuals, that they're going to drive the cluster and bias the clusters. So we do downsample for the actual cluster definitions. We've found that downsampling, when we apply mask, only reduces power. So there is no need to downsample.
And then, thank you. And then the second question from Neil Warner, did any of your cases have monogenic forms of disease that might help you with respect to directionality for the cell types, IPEX and FOXP3 as an example?
So not here. No, we didn't have. Unfortunately, I think that's a great suggestion, and it would be a lot of fun to do that. But no, we didn't have the chance to do that here.
Great, thanks.
Okay, thank you. Any other, before I dive forward, any other questions? Okay, I will charge ahead. So now I'm going to talk about covariant neighborhood analysis. So up until now, I've been talking about cluster abundance methods, which I think many people in the field are using. But an alternative approach is testing what we call neighborhoods. So rather than testing these clusters, we wanted to ask the question, is there a benefit to focusing on individual neighborhoods? And the idea of a neighborhood is that you're kind of defining a fine scale structure around each cell. You're finding for every cell its kind of closest friends. And then what you can do is you can ask, is that little tiny neighborhood associated with phenotype? And as you can imagine, this has some advantages and disadvantages. And the immediate advantage you see is that it allows you to really say, where in this like massive space of cells am I, seeing like that needle in the haystack? Am I able to find that small little neighborhood that's disease associated? And the way we define our neighborhoods is we use a random walk. So as you know, when you deal with single cell RNA-seq data, what we often do is we have a whole bunch of cells. We define a graph, a nearest neighbor graph. And you can actually exploit that same nearest neighbor graph to define your neighborhood. And what we literally do is for every cell, we do a random walk, take a bunch of steps. The number of steps varies a little bit depending on the size and quality of the data. And then you see if you end up in a particular cell. This red cell is your anchor cell. And if you end up at that cell, you get assigned to that neighborhood. And of course, this is done probabilistically. So depending on what the probability that this cell ends up at this anchor cell, that probability defines the extent to which this cell belongs to this neighborhood. So this might seem kind of intuitively very appealing, but there's some practical challenges that will be obvious to many of you, which is that a neighborhood may be too sparse to represent all the samples, right? So you can imagine here, if you have this neighborhood with a small number of cells, every sample may not be represented in this neighborhood. Some samples may be missing. And when you do your statistics or you do some kind of a statistical test to see if this neighborhood is associated with case control status, you might get some really weird statistics because there might be a lot of samples missing. Another challenge is multiple hypothesis testing, right? So the way I've set this up, if you've got a data set of 100,000 cells, you've got 100,000 neighborhoods. So that's a lot of tests, and that might reduce the power to find what you're looking for. And there are now multiple neighborhood-based methods out there in the single cell world. And let me tell you how we solved what we thought of these challenges that were bothering us. how do we manage the sparsity of these neighborhoods, and how do we manage the large numbers of these single cell neighborhoods? So the first thing we recognize is that single cell neighborhoods may correlate. So you could imagine that there's groups of neighborhoods that are more frequent in some individuals and rare in others. So for example, if you can imagine that somebody who has more Th17 T cells will cause lots of Th17 T cell neighborhoods to be expanded. So there is a kind of a correlational structure there. And of course, neighborhoods that are just adjacent to each other in transcriptional space, they may actually even share the same cells and not just the same pathways and the same biology. So again, you're going to have a kind of covariant structure there. So the assertion we're making here is that focusing on subsets of neighborhoods that track together may reduce the multiple hypothesis testing burden and buffer the sparsity. So rather than testing every neighborhood by itself, we can kind of group neighborhoods together that are behaving similarly in the cohort. Judy, did you have a question?
There is a question from Shika Nayar. What cell intrinsic attributes are used to classify cells in the neighborhoods during the random walk, and how are cells then prioritized accordingly?
Yeah, so for the random walk, we're not really prioritizing cells at that point. We're simply doing a nearest neighbor graph, right, where cells are assigned to be neighbors to the cells that are most similar to themselves transcriptionally. Most of the time, we're simply using straight up RNA-seq profile for a cell. In some cases that I'll show you, when we're, for example, looking at site-seq, we're using kind of an integrated vector, which captures both the protein and the RNA data. So there's an important choice there, depending on what you're doing. I know many of us are now working with multimodal RNA and ATAC data. So it is important to think a little bit about what that metric is that we're using to define this nearest neighbor, the nearest neighbors for every single cell, because that's ultimately what determines the neighborhood. Did I answer the question?
Yes, apparently. Thanks.
Okay, great. Okay, so this work that I'm presenting here is work from Yakir Reshef, who's an incoming rheumatology fellow, and Lori Rumker, an MD PhD student in the group. And so our approach was to define these local neighborhoods of cells and then use correlation across samples to define kind of relevant groups of neighborhoods. And I'll try to walk you through the method to give you an intuition. So the idea is you have all your cells here, and you can imagine that you've done a nice study. You have hundreds of thousands of cells from 50 samples, maybe from PBMC or from tissue or something like that. You've put them all into one nice map. You've defined your nearest neighbor structure. And now you do your random walk, just like I showed you before, and you start defining these small little neighborhoods all over your data set. And now here, I've just illustrated 5 example neighborhoods, A, B, C, D, and E. And now what I can start doing is I can start looking at different individuals. So you can see here in individual 1 and individual 2, they all seem to have a lot of neighborhood B, D, and C, right? B, D, and C. In contrast, you can see that there's some kind of different structure here for individual 3. Individual 3 seems to have neighborhood A and E. So maybe those neighborhoods are correlating, and individual 4 looks maybe a little bit random. So what you can do now is you can take all these neighborhoods and define a neighborhood abundance matrix. So each letter here represents that individual neighborhood. So you see here, for example, just want to make sure I get these letters right here. So you can see that that neighborhood B here is represented in sample 1 and sample 2. Neighborhood D is also represented in sample 1 and sample 2. So you can build this matrix, which is literally a count of which cells are in which neighborhoods and in which people. And now to capture the covariation structure, we do what we always like to do, which is we apply principal components analysis. which then allows us to define these principal components, which capture the major trends of different neighborhoods. And now we can simply use the principal components to test for phenotypic associations. And as you can imagine, we have many fewer principal components than we have neighborhoods, but the principal components capture the major shared structure across the data set. So how do you assess an association to a phenotype? So it's a simple linear model. You test for global association. You ask if these principal components, which capture this neighborhood structure, is associated with your phenotype. And so again, just like any linear model, you have your outcome. So if you're interested in something like RA, like I am, your outcome might be, do you have RA or not? Ones and zeros. And then in your linear model, the most important thing is you've got your principal component effect, and you've got those principal components from your neighborhood abundance matrix, from your NAM, those PCs we were talking about. And now you're simply asking, does this model predict to any extent what the outcome is? And of course, again, very important, you have the opportunity to include covariates such as age and sex and batch effects or whatever you think you need to include. One of the key questions, of course, you might be asking is, how many PCs should you use? And that's a really tough question. Any place where we use principal components, you always wonder how much should you use? And obviously, including more principal components has the advantage of capturing more of that rich neighborhood structure. But at the same time, the more you add, the more you lose that benefit of the dimensional reduction of the principal component. So what we do is when we have this model, we try a range of PCs, 5, 10, 15, 20, and so forth. And then we try this range of PCs, and then we test the association with permutation. So that gives us the ability to try a range of principal components and pick the optimal one, and also not overfit because we're permuting the outcome to test for significance. So that's the general approach. Before I move on, any questions on that?
A question just came up. Are the neighborhoods non-overlapping? If so, how do you define their limits?
Yeah, so the neighborhoods are overlapping because, as you can imagine, a cell can walk to multiple anchor cells. So we do allow them to overlap. And the way we deal with the fact that they overlap is through this neighborhood abundance matrix PCA structure. Because what happens is neighborhoods that share cells end up being correlated together, and that's picked up by that NAM structure. Okay, so what about power? So obviously the first question you might be thinking of is that this method is a little bit complex. It includes a whole bunch of machinery. It's potentially more complicated than just taking a cluster and testing for abundance. It obviously offers you a lot more flexibility to get the exact boundaries correct. So the risk is that you might lose power under some circumstances. So the ideal result is that it maintains power if a cell cluster is driving the association. You might imagine that this method is like going to be less good at detecting that signal. Hopefully it's equivalent. But you might imagine that if you have a more complicated association structure, that this strategy will be better at detecting it. So we had some real data. We had single cell data on 500,000 T cells from 250 individuals. And what we did was we kind of ran some simulations. We assigned a phenotype based on a particular signature. So let's say we thought a cluster was driving the signal in our model. We would literally quantify how much of that cluster is present in each sample. We define that as a phenotype and introduce some noise and see how well we do. Judy?
Yeah. From Harrisman-Carr, how is this different from a neighborhood-based clustering algorithm?
Yeah, so it's different in the sense that so when you do cluster abundance, for example, you often do use neighborhood-based clustering. Here, maybe what I'll do is, as I move on, it'll become clear how this is different. This basically allows you to find structures in your data that aren't well represented by a clustering algorithm. A neighborhood-based clustering method still partitions your cells into a bunch of discrete groups. which
Okay,
might.
and just as you go on, Shauna Cooper, how stochastic is this? Fairly reproducible with multiple runs or critical to set seeds?
Yeah, so the only stochastic part is the permutations. So it's pretty robust. So basically, there's no random seeding or anything like that going on here. But if you do enough permutations, your p-values are robust. So, so this is the first kind of power analysis that we did. So imagine that you have a causal cluster. So this is that discrete cluster that you have in your UMAP space. So here I've illustrated in red. And we did a power analysis using both cluster abundance methods and CNA. And very gratifyingly, they're equivalent, right? So you might have expected here that the abundance-based method would do better because abundance-based method is trying to find case control differences in a cluster. CNA is totally flexible. It's doing fine. This is where it gets interesting. So now in the model on the left, where a single cluster is driving, your power is equivalent. Now let's imagine that you have a different situation. Your signal is not being driven by a single cluster, but rather it's being driven by some kind of complex structure. So in the world of rheumatoid arthritis, fibroblasts, for example, we know that there's a polarization of fibroblasts with notch signaling. So in that case, you have an expansion of some populations, a contraction of other populations. Now you see that if you took the space and partitioned it with clusters, you'd be looking at something really weird. You wouldn't be able to see what you want to see. And here you see the power advantage, where CNA is having a power advantage over the cluster abundance method. Now something even harder. Imagine that you have a single cluster, but within that cluster, some of the cells are expanded in your cases and some of the cells are expanded in your controls. Now when you look at that, you see that there's a power advantage with CNA. CNA is able to find the signal. Cluster abundance methods struggle a little bit because obviously all of these things live, presumably in one cluster, or maybe they're being split into a couple clusters. Judy, there were some questions in the chat box. Oh no, they're just statements of gratitude, I think.
Yes, correct.
Okay, yeah, you're welcome. So that's great. So that power just tells you, like, are you able to find something when there's something in the data? Another important question is, are you able to find the right thing, right? Like, you don't want to just be able to say that you're finding something. So signal recovery, are we finding the right cluster? So we have a particular signal recovery metric. Here's the simulated signal. Here you're kind of mostly getting the signal right. Here you're not getting this, you're kind of finding the association, but not getting it totally right. So we have this kind of signal recovery metric. Now look, when you look at CNA versus clustering, when it's a cluster driving the signal, both are doing about the same. Now looking at these other two models, you see this is where there's an advantage to CNA. It's better able to define the cells that are driving your structure. I'll show a practical application, and in the interest of time, maybe I'll speed along here. This is a data set on sepsis, where there was 29 cases, 36 controls from Nir Cohen's group. There's a total of 103,000 cells. We applied these different strategies. So if you apply CNA, you see this very complex structure of associations. The original authors reported this MS1 cluster, which we found to be only kind of nominally significant when you do a cluster only. The CNA, the cells seem to be associated with a particular pathway. The cells that are more expanded are being driven by RAC1. The cells that are a little more depleted, less so. But to me, this was the really interesting thing. When you look at those individual clusters and you look at what's associated and what's not, you can see that there's a lot of heterogeneity. So this MS1 cluster has cells that are strongly associated with the phenotype, other cells that aren't. Look at this MS4 cluster, you see cells on both sides. So it's exactly that model where within that cluster, there's some cells that are protective or some cells that are depleted in cases, others that are expanded. Very quickly, we did a study on tuberculosis. This was work with Socios and Salud in Peru. So maybe just in the interest of time, we looked at T cells in these individuals and simply asked, is there a T cell population that's depleted in individuals that when infected with TB get sick and progress to being sick versus those people who when they get infected, they stay in a latent state. So as you can see, our case control comparison was people who developed primary TB versus people who had latent disease. So we obtained data on 125 cases, 125 controls. We sorted memory T cells. All these individuals were from Lima, Peru. It's work done with my collaborator, Megan Murray. And then again, maybe in the interest of time, I will skip this. This was a situation where we were doing a site-seek study. So we recognized, to understand what was going on with T cells, we would want to look at both RNA expression as well as surface marker antibody expression. So looking at both together, and the analysis I'm about to present was all done by Aparna Nathan, looking at RNA and protein, integrating that data together using canonical correlation analysis. So we had basically a linear vector that captured the dual signal of RNA and protein for every individual cell. So we had this kind of rich T cell cluster. We defined our clusters, as you would imagine. We saw CD4 versus CD8 differences very clearly. We were able to see, CD62L positive and negative cells capturing the effector versus central memory T cell gradient, CD25 positive Treg cells, and many other T cell clusters, and I won't get into all of them. You see that there's a lot of Th1/17, Th17, other related clusters. So we went back to mask, right? So when we first started this project, all we had was our old single cell cluster abundance method. And sure enough, we saw some very interesting associations with age and sex. So we knew we had to use them as covariates. You can see some populations are depleting dramatically with sex. With age, there's sex differences in T cell populations that are well known. Now, when we control for that, we found that there was this population C12. You see there's a bunch of other nominally associated populations, but C12 was about 20% deficient in those individuals that progressed to TB. It looked like C12 might be a Th17 population. Again, in the interest of, this is actually an example where it was known that RORC mutations, which are essential for the Th17 pathway, are deficient. And when it's people who have mutations in RORC, they're more likely to get MTB infection. And I'll just skip this because I know I want to have some time for discussion. When we functionally characterized these cells, we saw that these cells were secreting IL-17 and interferon gamma. So they're a Th17 mixed population in terms of what they're doing. And we did some studies to see if they were responsive to TB antigen, and indeed they were. So we thought we had this population of T cells that were critical. We had a bunch of surface markers that define them. They were a very specific cluster. And to summarize, we believe that individuals have a variable capacity to mount a Th17 quote C or C12 state. Total seq, or this RNA combined with protein marker strategy, helped us find this population. And we did this all with this cluster abundance approach, using a data set of 500,000 single cells in 250 individuals. Now, what if we had covariant neighborhood analysis, which was developed at the same time we were working on this project? Would we have found something different? Would we have found something better? And again, this is that spectrum of different T cell populations on the left. And I've labeled a lot of these Th1, Th17 populations. C12 in particular here is that one I was talking about that we found to be depleted in TB. Now, this is where it gets interesting. Now, when you apply CNA, you do find an association with that C12 population, just like I told you, there it is. But you find that all these adjacent clusters, which were only nominally associated in our initial analysis, are also associated here. So we see a much bigger spectrum of cells. And guess what? All of these cells, what do they cover? They're all other Th17, Th1. They're capturing that same general spectrum of cells. So biologically, we're living in the right space, but we're getting a better picture of what's going on. And we're also we're a better powered method, so we're able to find some populations that are expanded as well. Here's another bunch of variables. This large data set allowed us to look at many things. So we can look at TB progression, as I just showed you, but we can also look at the age associations. We can look at associations with seasonality and association with ancestry. And in each case, CNA was a better powered method that got us the contours of which cells were driving the association in a much more effective way. So I'll conclude the talk and take questions. So CNA, I think, balances the granular resolution of sparse single cell neighborhoods with the covariant structure of neighborhoods across populations. So we're able to test at a much finer resolution, but we don't get killed with multiple hypothesis testing or sparsity because we're taking advantage of this covariant structure. It's based on a flexible linear model that can be easily adapted to many circumstances. It offers power advantages of cluster abundance methods, and it can be used to define associated subsets of cells, even when those cells don't respect the boundaries of a cluster. I showed you some power analyses, which showed that it was better powered than a cluster abundance method. I showed you a sepsis data set and a TB data set. And most of these results that I showed you today are in blood, but I'll share with you that we're doing analyses in synovial tissue as well, where CNA has shown equal promise. So with that, I just want to thank Yakir and Lori, who co-led the development of covariant neighborhood analysis, Chamit, who led the development of MASK, and Aparna, who led that nice T cell study in the end, and also worked on many of these methods along the way. And with that, I will stop and take questions.
Thank you so much, Shomal. That was just absolutely terrific. So folks can, oh, here we go. From Shifa. Have you tried this approach with ATAC-seq?
No, we haven't yet. So we haven't yet gotten our hands on a data set that's large enough, but I would imagine there would be some very interesting questions there. We have seen that when we look at single cell ATAC-seq data, we do see different structure than what we see with single cell RNA-seq data. So we are really excited to try this approach once we get our hands on a cohort.
So anyone can just, I think we have a small enough group that anyone can just speak out if they don't want to type. Oh, yes, Yu Zhao, go ahead.
Hi, hello. Thank you for your talk. I have a question regarding to the cytotoxic CD4 T cells that you showed in the paper. You didn't mention the function, but we have also noticed that in some other autoimmune or infectious diseases. So in this specific tuberculosis data, do you have any deeper insights of their function, and how do they differ from the CD8 cytotoxic T cells?
We don't, but we've been struck by that too. And in fact, we've had reviewers comment and say, "Oh my God, you have so many CD4 cytotoxic T cells. This has to be a mistake." But I don't think it's a mistake.
I have exact same feeling when I look at some other data, and we found a lot of cytotoxic CD4 T cells. So and also even in the some COVID patient data. So it's definitely something novel, I would say, but unfortunately we don't have a very deep understanding. So another relevant question is, I read a review paper that you published on transient immunology, talking about the TCR barcoding, right? So in the RA and this tuberculosis blood, I'm not sure if you have already seen the TCR sequence combined with single cell approach, and can we interpret some antigen presentations into the potential clonal expanded T cells?
So, I think clonal expansion, I mean, I guess I'd be interested to hear everyone's experience here. We've started to look a little bit at single cell TCR and single cell RNA-seq, combined data. And, it's a tough area. I mean, one, I think like everybody, we see a lot more clonal expansion in CD8 T cells than CD4 T cells. And the idea of using TCR as a barcode is probably a much better idea for CD8 T cells than CD4 T cells. We've also been looking at TCR sequences in synovial tissue. I had personally really hoped to find lots of clonally, I had maybe a model in my head when I was a rheumatology fellow many years ago that, there's a citrullinated antigen. That citrullinated antigen is stimulating Th1 T cells, and they're expanding like crazy, these CD4 T cells, and that there's a lot of TCR clonal expansion. But actually, when we've looked in the data, we don't see this. We see a lot of CD8 TCR expansion, but there's a lot of CD8 TCR, clonal expansion in the blood. But what we see in the tissue is much more limited. So to me, it means probably that there's a lot of different citrulinated antigens. There's a spectrum of antigens, and the CD4 T cells aren't proliferating as much as the textbooks would have us believe in autoimmunity. But this is a yeah, I'm sure you guys have thoughts on this because you're probably seeing different or similar things.
So I'm going to read out two last questions from Ken and Eric. Ken, my question is, what do you think is a good set of validation performance metrics for the end user to look at for choosing among various neighborhood methods, SIDAR, Milo?
Yeah, I think what's really useful, I mean, this is a little bit like some very maybe basic advice, but one of the things we love to do is just to permute the case control status and run the method and just see how it does. I don't want to be calling methods out, but there's a lot more false positivity with these methods than you would think. So to me, that's the key thing. So if you permute your results and you start seeing a lot of neighborhoods associated, you know that you're in trouble.
And then from Eric, is there a way to extract an interpretable gene pathway signature that is driving the neighborhood classification through the principal components or otherwise?
Yes, there is. So, it's not like many things with sort of weights and principal components. It's not the easiest thing, but basically what you end up doing is you can recombine the principal components with their weights. And then what you end up doing is getting a weight for every single cell. And so then what you can then do is you can ask, how do those weights correlate with individual genes, individual protein markers, or whatever feature you want to look at the cell, including, pathway scores. So that's how we've been doing it.
Well, I think we've had a wonderful set of questions, wonderful seminar, great data. And as you said at the beginning, Soumya, we're at the beginning of a revolution. I think we're just at the beginning. So I think it's a lot more to do, guys.
I think you're totally right, Judy. Thank you so much.
Thanks, everyone.